Hey guys, so we have been learning about the cell membrane recently, and what we're going to do today is a lab that demonstrates some of the four main concepts that we've been talking about. So if you can remember, that first concept is that the cell membrane is fluid and flexible, so that means that it doesn't stay rigid and stand still like this. It kind of does a little bit of movement. It's really fluid, okay? And then the next concept is that it can self-repair. The cell membrane can self-repair. So if it has had any damage done to it, if it breaks open, it closes back up. It can self-repair. The third one is that the cell membrane has membrane-bound organelles. And then the fourth concept that we're going to talk about today, and we're going to be able to see in our lab, is that the cell membrane has those membrane proteins, those special proteins, and what they do is special functions like maybe allow some things in and let some things come out. So that is what we are going to be doing in our lab today, and so let me get down to it. Alright, so you are going to need a few things at your lab table. At your lab table, you are going to need 5 bendy straws. You are going to need a measuring cup. Hopefully it doesn't already have liquid in it like this one. If it does, just pour it out. You don't need it. You're going to need a pan, a little shallow pan that I've got for y'all. You're going to need like 4 inches. This is way more than 4 inches, but you're going to need about 4 inches of thread. And we are going to need 900 milliliters of water. We're going to need 100 milliliters of dish soap. And we're going to need about 25 milliliters of this corn syrup. Okay, so the first thing we need to do is get our mixture together. And so the first thing that I do is, because I only have a measuring cup that goes in increments of 100 milliliters to 150, and I need 100 milliliters of the dish soap and 25 of the corn syrup, I'm going to pour out 100 milliliters of our dish soap here. And remember, whenever you are measuring things accurately, you need to be at an eye level to make sure that you don't get more than 100 or less than 100 in the soap. It's looking like I got about 100 when I got eye level there. And then what I'm going to do is I'm just going to add this corn syrup and get it about halfway between the 100 and the 150 mark, because this is just a biology lab. It doesn't have to be exact science. And if we need to add more later, we absolutely can, because science is all about trial and error. That might be a little over 150, but I think we'll be okay. All right. And then I'm going to go ahead and pour this into our tray. And now the next bit that we need is 900 milliliters of water. You'll see that this measuring cup does not go to 900 milliliters. So I'm going to fill it up to 500 first, and then fill it up to 400. And I'm going to have those poured into our tray as well. Just about good there. So that's 500. And now I need 400 to make 900 milliliters. And our next one, I'm going to take these bendy straws and I'm going to make our frame here. So what you need to do is, I like to take the short end and put it on the outside. No, on the inside. Outside, inside. I don't know. One side is easier than the other, but you're just going to make a small 4-sided frame. This one's a little difficult. All right, and boom. You should have your frame that you can now dip into the solution. Now, before you do the actual lab, I do like to mix the solution around a little bit, get it all mixed up to where the bubbles are kind of easy to make. So I would go ahead and do that. So it's all good and mixed up. Go ahead and also tie your thread. I have nails, so it's a little difficult, but it'll be fine. We got it. Boom. Yep. You're going to want it to be about this size. So our first concept is that the cell membrane is fluid and flexible. So we know that cell membranes are not rigid, they're not just standstill. What they do like to do is bend and flex, and they adapt to the changing conditions around them. So what you need to do first is you're going to submerge your bubble frame into the solution and then slowly lift the frame out. So when you slowly lift that out, you're going to let the excess fluid drip down from the frame, and then you're going to tilt the frame back and forth, and you're going to observe the surface of the film. Do this basically. You're going to make it bounce up and down. Or you're going to... After that, you're going to carefully hold the frame by its edges. And what you're going to do is you're going to rotate the sides of those edges. You're going to push in the edges. You're going to pull out the edges. And what you're going to be able to see is that the bubble moves with the frame, right? So this goes hand in hand with our first concept of the cell membrane is flexible and fluid. Just like this bubble, the cell membrane will adapt and it will be fluid and flexible along with its changing environment. And this is due to the attraction between the phospholipids. We know these phospholipid heads are hydrophilic, that's right, we have hydrophilic heads, and that means that those tails are going to be hydrophobic. It's not going to like water, it's going to be hydrophobic. So for this second experiment to explore this concept, what we need to do is, again, we're going to submerge the bubble frame into our solution. We're going to slowly lift the frame from the solution, and we're going to allow all of that excess solution to drip off from the frame. Then you're going to cover the surface of your finger. You're going to take your finger, you're going to dip it into the bubble solution. You're going to make sure you get all of your finger kind of coated in it. You're going to want to make sure it's lathered, okay? And next, you are going to slowly push your finger through the bubble frame, and then you're going to pull it back out. The bubble film shouldn't pop. It should repair itself. And it didn't pop. So try coating if you want to. You don't have to. What you could do for a little extra experiment is coat your whole hand in the substance. Can we put our whole hands through, our entire hand through this bubble frame and it still repair itself? Try it out. Like the soap bubble, the cell membrane can repair small tears in the phospholipid bilayer. And the third one is that eukaryotic cells are going to have membrane-bound organelles within itself. So like the mitochondria and the chloroplast and those smooth ERs and the rough ER and the Golgi apparatus, remember how those are different organelles within our cells? Well, the mitochondria has its own membrane around it, and that is a membrane-bound organelle inside of eukaryotic cells. Alright guys, our fourth and final concept that we're going to be doing in our experiment lab today is that the cell membrane is going to contain proteins within its membrane that has a method of transporting these larger charged particles that we talked about earlier. Well, they have to use proteins. They have to use these protein channels to push them through the membrane because they're a little bit too big to just go through it on their own. So if you follow these steps, we're going to be able to see that. So what we're going to do first is submerge the bubble frame into the solution. Go ahead and slowly lift the frame from the solution, and you're going to allow excess liquid to again drip from the frame. You're going to dip it into the bubble solution. Make sure it gets the bubble solution all around the thread, and then we are going to take it and put it carefully into the bubble frame. Now if your bubble frame pops when you do this, you need to start over. And it might be beneficial to have one person hold the bubble frame and another person put the thread in there. That might help a little bit. To get that thread into the bubble frame, you are going to take a pencil or something sharp. If you've got a pencil, that'll do. And you're going to poke it inside of the circle of the thread. Whatever circle is there, just pop it into the circle of the thread. So next we're going to take our pencil and we're just going to pass it through the membrane. This is going to represent charged molecules passing through the cell membrane using channel proteins. That's it. Alright, I hope this video was very beneficial and helpful for you in doing this lab either at home or maybe in your own classroom. I hope that it helped you show those 4 different concepts of our cell membrane and that you learned something today. Have a good day.